Welcome. I'm Liz Taylor, ocean explorer, naturalist, and entrepreneur.
I'm Sylvia Earle, National Geographic explorer, founder of Mission Blue, and I am an ocean elder. Thank you, ocean elders, for sponsoring this program.
And this is our show Dive In, where we host informal and open conversations with the ocean community from around the world on topics of wonder and interest. As we get through the conversation, please put your questions in the Q&A box and we'll answer as many as we can towards the end of the program. And today, a special thanks to Patagonia for hosting the space for us today. We're not in the home library in the normal spot.
Sausalito.
In Sausalito. So, but still, as we get started, we remind everyone that the world is
Blue.
blue.
All right.
Absolutely.
Blue.
It's part of the theme.
Yes. And today we are so excited to have Thomas Cronin joining us. Tom is a professor of biological sciences at the University of Maryland, Baltimore County, and he leads the Cronin Lab, a team studying visual physiology of invertebrates, especially those in the marine and estuary environments, and other vertebrates as well. And he is a recipient of the International Rank Prize for Optoelectronics.
Ooh.
Welcome, Tom.
Hi, it's nice to meet you all. Nice to be.
Yeah, I'm going to try to share my screen here so we can jump into your slides. But I wanted to say that in our home library, we have a wide array of books, lots of field guides with useful tips on how to recognize the animals that we encounter and the wild birds, especially, and insects. But it doesn't tell us anything about how those animals recognize us, how they see us. So I'm really anxious to dive in here.
Great.
The wonderful thing is the many variations on the theme of eyes, of vision, of perceiving through sight, through light, the world around whatever those critters are. It's not all the same.
Not at all.
Yeah, you all actually might have seen there's been a lot of publicity about a camera that was recently built that allows people to film scenes that to some extent reproduce what animals might see in those scenes. So that's something we've been doing a long time with visuals as well. But shall I start on tell you a little bit about animal vision in land
Absolutely.
and sea? So this very beginning kind of sets the stage for what I want to talk about. The two sides of the screen look very different from each other. And obviously the challenges of vision are much harder on one side than the other. So in the next slide, you can see a variety of the eyes that have evolved over time that cope with these different kinds of environments. Most of the eyes you see here are eyes of animals that live on land. But interspersed in between these, you're going to see eyes of different animals that live underwater. And you'll probably be pretty hard pressed to pick which ones are which. Stuck in here at various places are a fish eye and a shark eye, a cuttlefish eye, a bunch of crustacean eyes, some worm eyes, and others that live in the sea. So if you go to the next panel here, please, Liz.
Here we go.
The eyes that are called lens eyes or camera eyes. These are the kind of eyes we have, and these are the kind of eyes that most vertebrates have. So fish or marine mammals or diving birds would have eyes like these and use them underwater. But of course, in most cases, these are terrestrial animals. And these all form an image the same way that the classic camera forms an image on a sheet of film or a CCD array in a digital camera. They just, the lens focuses the visual world onto a sheet, and that then contains photoreceptors, and they pick up the world. Let's go to the next set of eyes then. So one, then go down again, and we'll have left behind one more hit. We'll have the compound eyes. So this is a totally different design. Here, each eye is not a single eye with a single lens, but instead there are hundreds or thousands of individual lenses, and each lens feeds a little single set of photoreceptors. And so it still forms a visual field, and it still has a visual world, but the world is sampled with a completely different kind of eye. And although there's fewer of them in the picture here, there's actually many, many more of these in the world because all the insects have these, and virtually every crustacean has these, and some other odd animals, like the animal on the far left is a tube worm, has eyes like these. So they pop up again and again in nature. It's not really clear why they're so popular because they're not really a great optical design, but they have been incredibly successful. And among the animals that have these eyes are mantis shrimp. So the eye in the middle is a mantis shrimp eye. There's some other mantis shrimp eyes here too. And we'll talk about these as we go forward.
One more.
And the last panel is just, there are some eyes that aren't lens eyes or compound eyes. If you look carefully at this little bloom worm, this belled worm, you'll see that there's little dots around the radials, the gill fan. And those little orange dots, which are barely visible, are eyes. This animal, even though it has hardly any brain and it lives in a tube, doesn't even have a head, it still has a couple of dozen eyes that it uses to see the world. So eyes are everywhere.
Amazing.
So back to the terrestrial world. So what we have here is a beautiful terrestrial scene. The Blue Ridge Mountains out here in the east, looking out to the west in the evening, and you can literally see for miles and miles. You can see ranges of mountains that go off into the distance almost as far as there's land. If you look straight up, or if you look up in the direction of the sun, you'll see the sun, which is now 93 million miles from us. If you wait till nighttime and you look out, you'll see stars that are hundreds of light years. You'll see galaxies that are millions of light years from us with your eyes. You don't even need a telescope to see them. Seeing distance is essentially infinite as long as something is bright enough. But when you go underwater, next slide, the world's not the same at all.
No.
On the best water I've ever seen, I've never dived in the open ocean, but I've dived on pretty clear reefs, was maybe 30 or 40 meters was as far as I could see. This scene is taken at Heron Island on the Great Barrier Reef. It's really semi coastal water, and so the water there is pretty blue and murky. And the reason it looks basically greenish is because, or green blue, is because water is a blue filter. We think of water as being clear. You look at it in the glass, it looks just as clear as the glass does. But in fact, if you look at a swimming pool, you can see that a swimming pool is filled with water, but the water looks blue. And you look at the ocean, it looks very blue. It's not because it's reflecting the blue sky. It's not because it's scattering blue light like the sky does. It's inherently blue stuff. Water absorbs red, it absorbs short wavelengths, it absorbs green and yellow, and the only thing that gets through is blue. So as you go deeper in water, like the sultry fish here, the red of the fish disappears, and all you have left is the gray, because red what does water.
It goes so fast too. It's really surprising.
Yeah,
it's really surprising. You get into water in the ocean anywhere, and you see a few bright fish, but you go down, 30 feet or so, and
turn on your light though.
Yeah, you need the light.
Yeah, and as animals are further and further away, they're that color. So here was the scene we started with, and if you punch the button, you'll see that with a proper visual system, this is the same photograph, but you can extract the information you want from it. And this is what marine animals have to do all the time. They have to contend with a world that's very low in contrast, often very dim, and very limited in color. Typically in most marine waters, unless you're very near the surface. This was not very deep, so you can see that the squid themselves do have some reddish coloration.
Yeah.
But the low contrast has been improved by a much better visual system. So this slide just shows that, of course, the visual world of animals changes radically as you go deeper. So near the surface, it's pretty bright, almost white light, or at least bright blue white light. You can have nice eyes matched to that. But as you get deeper, down to 600 meters, down to 800 meters, and finally down to 1,000 meters, there's no light left at all. Animals still have eyes there. They're not using those eyes to see sunlight anymore. They're now using it to see bioluminescence, which I know you've talked about before.
Right.
The animals of today's talk don't have bioluminescence, so they don't use their eyes down there at all.
It's so funny.
These animals obviously are very different.
Yeah, and so many of them are really brightly colored.
Yeah, well, as you get down deep, the colors you see are either transparent or black or red, because red, like we saw in the last slide or earlier on, in the deep sea, red is invisible. There's no light, there's no red light, so you don't see red things. They're just black. This slide just shows you the two major kinds of eyes again, the chambered eyes like our eyes, with a lens and a retina, And the compound eyes, like the eyes of mantis shrimps and crabs and lobsters and shrimps and insects as well, which are built from many, many different units. So the next few slides just show a few examples of some odd eyes. These are the strangest eyes you might expect. This is actually a jellyfish. It's really a cubozoan or a cubomedusan.
That's a great
In
word.
life, this little guy, this particular one's only about maybe a centimeter or a half an inch across. It's quite a small animal, but it has 24 eyes.
Man,
The eyes hang down in those little, if you look at the picture in the lower middle part, it shows a rhopalium, which is the a little sensory structure that hangs down from the bell. There's 4 of these in one of these animals, and each one contains 6 eyes in it. 2 lens eyes. They're the eyes that you see in the picture on the right with this that look like big circles.
yeah.
2 pit eyes, which look kind of like slanted eyes, and 2 slit eyes, which look like little eyes with lines drawn through them. So this animal, which again doesn't have any brain, has only a simple nervous system, is carrying around 24 eyes to see the world with. So eyes are found in the unexpected places. Here's a polychaete worm. Some of you have seen these, I'm sure.
Yes.
They call it an alcyopid. It has gorgeous camera eyes. The eyes are based the same plan as our eyes, but it's carried on a segmented worm in the open ocean. It's quite a surprise.
It is really surprising. And these are I love these guys.
Flying eyeball.
So
Yeah,
cool.
really. There's lately been a lot of fuss about how some of these ostracods are bioluminescent. They form displays in the Caribbean. They drop a trail of bioluminescent dots in the water, but those animals have compound eyes, like most crustaceans. This is a deep sea crustacean, familiar to Sylvia and Liz. And this animal has eyes that are built with reflectors, like the reflectors on a headlight of a car, but the headlights reflectors, of course, work to project a beam out. And these reflectors work to collect light. So the animal has very, very sensitive vision. And in fact, this animal on optical principles has one of the most sensitive eyes in the whole animal kingdom. But it's basically this little globe with these 2 bright sort of golden reflectors in the middle of it, looking for bioluminescence.
They're just such interesting animals, and the freshwater ones are really cool too. I one time found some just occurred in this pan of water, and I was like, look at, we've got these ostracods.
If you walk
So
along the
exciting.
beach, especially in the tropics at night, on the edge of the beach, you'll often see bright blue flashes. They're often ostracods that have washed up.
Now I gotta go look for some.
And just to point out that eyes have been around a long time. This is a fossilized compound eye of a trilobite. We worked on this many years ago. I had a student who wanted to work on fossils, so we decided trilobites would be a good choice. And you can actually recreate the visual worlds these animals had by mapping out how their eyes sampled the world. That's what we're seeing here, is that the green dots show the visual world to the animal's right, and the red dots show the animal's world to its left. You'll notice that vision overhead was very limited, and so they wouldn't see approaching predators very well, which is kind of worrisome, I would think, for a little creature living on the bottom.
Yeah, that might be a little rough.
Of course, they went extinct, maybe probably because they missed
That's
seeing
right.
important things.
And the tubular, yeah.
These are strange eyes that you may have seen before. They're called tubular eyes. They're eyes that are not shaped like a globe, but instead they're shaped like a bullet or a tube. And they've evolved in, there's actually some compound eyes animals that have these, but here's 2 lens-eyed animals, a deep sea cockeyed squid called Histioteuthis, with 1 big eye that looks up and 1 little eye on the other side that probably looks down and around.
Wow.
The upper eye is a tubular eye. It just sees a little patch of light overhead. And then here's a fish, Stylophorus cordatus, photographed by Edie Witter, that shows the bullet-shaped eyes that it has, with a lens in the front and a tube shape that leads to a retina in the back.
So
There's some
interesting.
very interesting tubular eyes out that have been found in Macropinus, which was found out in the Monterey Bay.
Oh yeah, is that
15 years ago.
the barrelhead fish? That guy?
Exactly.
Yeah.
Now, something we don't think about too often is one way to enhance vision underwater is to use special wavelengths. So here you see a reef scene taken with a camera that had a green filter on one side and a UV filter on the other side. It was two Nikonus cameras back in the old Nikonus days. So two film cameras, but you can see with the green filter, it's hard to see all the little fish that are there that are sitting above the reef. But with the ultraviolet filter, everything stands out because the UV world in shallow water is very bright. And so you can see anything that stands out against it that's nearby looks black and is easy to find. So
Yeah,
it's great for predation on small animals. And a lot of fish use these to find prey. Okay, so now we're into the stars. This is a gorgeous animal. It's a Clinodactylus terratensis, which is Clinodactylus terratensis, which is an Indo-Pacific species. It's kind of posing for the camera here, but it points out some of the really spectacular things about mantises. There's beautiful colors. They're sort of strange shape. They're big front ends with the heavy devices that are used for predation. And of course, the strange little eyes that sit on the top of the head here. And we'll see that mantis shrimps you probably know.
Yeah.
They probably think we have strange shape.
That's probably true.
It works for them.
They only see us with those sort of, a mask on, right? So they're like, what kind of eyes are those?
Yeah.
But I've taken
My friend Mike Land, who's recently passed away, calls these shrimps from Mars because they have so many strange features.
They do, and I've taken care of these guys in aquaria before, and we always have to put them in a plexiglass tank because they have the capacity to actually break the glass on the tank, and then if you go in there to clean the tank, you always kind of watch your digits because they can break a finger.
Yeah, they can give you a nasty punch, or they can give you a nasty infection if they inject, some algae and things into your finger too.
Yeah, but I think in general they tend to be not too agitated, thankfully.
No, they actually they're lovely to have in aquariums because they're usually doing something, so they're fun to watch.
Yeah,
They're curious. You can.
So most of these photographs were taken by Roy Caldwell, a colleague of mine at Berkeley, who's a really excellent marine photographer. And so mantis shrimps come in two flavors. So this is one flavor. It's the spearers. They use their front appendage, it's called a raptorial appendage, as a snatching device, very much like a praying mantis does to catch, in the case of a praying mantis, to catch flies. But here they're, of course, catching other things.
yeah, he's really got
so that's why they got their name, mantis shrimps, because of this type. And these are the ones. If we were talking earlier about fish markets, this is the type you find in fish markets. Typically, they're bigger, and they're shallow water living, and they're relatively easy to catch. They're pretty slow and have a big tail with a lot of muscle in it.
Oh, poor guys. The slow and easy, right?
These mostly live in burrows. They dig burrows in sand and live in the burrows and hunt from them.
So just the eyes stick up. Is that what happens?
Yeah, the eyes are there on top looking up.
And the pinchies.
You wouldn't want to get your finger into that little catcher there. It would hurt.
Oh yeah, that would be
like this poor yellowtail damsel is finding out.
So what's sort of the distribution of the mantis shrimp? Are they kind of global or localized?
Yeah, they are global. But they're tropical. You don't find them very often in cold water. There are some cold water species. There's a species off the coast of California which you can find off of Catalina, for example, where they're really common. It's called hemisquilla. It's a pretty big one, and it's unusual for living in cold water because most mantis shrimps are tropical. This one's from Australia. There's a very similar species to this that live in the Caribbean that look essentially identical to this one. We do get a few in the Chesapeake Bay. They're of this type, the spearer type, but they have very, very boring vision, and that's the case. But almost all the species we've ever worked on live in the tropics, and most of them are associated with tropical reefs because they live in crevices and cracks or holes in reefs.
Yeah. Wow.
So that's what, like this guy. This is flavor number 2. This is the smasher, and if you look at it's hard to tell by this point, but you can see it's holding a snail shell. It's about to smash it to bits. And the part that it uses are those bright red objects that are to the right and left of the snail shell right now that look like a little bit like hammers.
Mm-hmm.
And as it snaps those out, it can literally break the shell into pieces in a
It's
couple
kind of
of
like this sort of thing, right? They just kind of like
exactly.
elbow.
It's like it hits with its elbows.
Yeah, yeah. I'll hit with my elbow too sometimes.
Yeah, and these guys live, they typically don't make their own burrows. They typically live in crevices or in holes in the reef. Well, these guys are pretty big. This species gets up to about the size of your hand, so it's a
Wow.
reasonably good sized animal. This is
So-
one of the ones that breaks aquarium glass.
Yeah, definitely.
You know, to our eyes,
So
they
it's
look
it's
so
called a smasher
small,
for a good
but they
reason.
look like creatures in the sea. With all of that gaudy, splendid color.
Yeah, the
Yes.
coloration is something that always catches the eye. This thing is called commonly a peacock mantis shrimp because of its beautiful color displays.
And but they're kind of fixed. It's like they don't they don't have like chromatophores or anything to shift color. This is kind of how they are.
Right. That's a really good point. They are not like many crustaceans, like many crabs can change color, or at least can get darker or lighter. Mantis shrimps are pretty much, they can change color over time. And in fact, if a mantis shrimp moves from shallow water to deep water, this species doesn't do that, but some do, and they actually take on different colors when they move into deep water that make them more visible to their conspecifics, so they can be seen in the same way that they would look at shallow water. So they often will get more contrasty colors that are appropriate for deeper water when they go deeper.
That's pretty interesting. And you think about something like this on like a really colorful reef. I wonder, like, having this much coloration, does it actually, in a way, kind of help disguise them from predators?
It does when often you don't see these colors displayed because they're on the bottom of the animal. So when you're looking at them as you're searching through the reef, you often won't see these colors. So when we hunt them, we're really hunting for the shape rather than the color of the animal. Although they're usually the ones we hunt are usually on sand, and you can see against sand they stand out pretty well. They actually they're not well camouflaged against sand. They're dark colors typically. Here's another smasher pair. This is Neogonodactylus ursidae. This is an animal we use a lot in our lab. It's from the Caribbean, so this is an Atlantic species here. The other one's in the Pacific. But you can easily see the smashers. They're all white in this species.
Yeah.
There's a female who's in her own burrow. She's sticking her head out, fending off a male who was trying to evict her from her home. And so she's smashing at him, and he's trying to defend himself.
Yeah.
She wins this battle, I'd like to point out.
Good for her, right?
Yeah. Notice another thing is that he's displaying. Could you go back to this for a second? If you look at what she's aiming at is actually his telson, the tail fan of the animal, which, if you see, has a lot of bumps on it. Those are actually designed to take the hit. So when she whacks him on those, it's kind of like hitting a cushiony area of the carapace, so the animal is not immediately smashed. It actually absorbs the hit.
Wow.
And so this is sort of a ritualistic fight. She hits him there, and then he comes back and tries to hit her back. But he tries to protect himself with this cushiony area.
Like a little shield, a little tail
It's
shield.
like a shield that
that's really cool.
that he puts out to protect himself. Exactly. Yep.
Amazing.
So as we've just pointed out, they're very colorful animals. Here's an array of different species, and you can see the color displays that they have. They're, right away when you look at these, you think these animals must have color vision, right? Why would they be so colorful if they don't have color vision? And that was what we started out to work on when I first started to work with these guys. My first experiments with these were back in 1983, so it's been a while. It's been a more than 40-year journey. And we still don't. We know they can see color, but we still don't quite know exactly what they do with it. It's been kind of disappointing after all these years. They do have extremely complicated color vision systems, which I can talk about a little bit. So that's shown in some of the slides coming up.
All right.
So this is a little video which might play or might not. Let's see how it works out.
If it goes, if it doesn't, I have a plan B. It says media not found, but let's see. Let's see.
Okay, I think we're good.
Can you see that?
Yeah.
Good.
It's running. So this is a video made by one of my grad students of this that same animal we saw earlier, the so-called peacock. Its real name is Odontodactylus solaris. But notice these odd ways in which it uses its eyes. It's not like any other animal, really. They can move independently. They can rotate. They can swing right and left. And you might notice these little dark spots on the eyes. There's sometimes you see one like you do now.
Oh, yeah.
Sometimes you can see two, and sometimes, but it's just right, which if we're patient, we might see that. You'll see 3 dark spots on the eye. And the important thing is the dark spot that you see is the part that's looking at the camera. So here you can see 2 in each eye. And the third one is really, really only comes out when the eye is looking right at you or right at the camera. So it's not very often that you get it, but hopefully he'll give you a shot. So what this means is, there you go, you can see it there for a second.
There it was.
That means these animals have triple vision in each eye. Each eye sees the same object in space. There's a nice shot. From 3 different places simultaneously, from the top half of the eye, from the bottom half of the eye, and from the middle of the eye. It's looking at the same position. And that's kind of shown, I think, in the next slide. Well, we can see a little bit of the inside there. Here's a nice piece of graphics showing, if you pop for the next bit, you'll see a bit of the retina of the animal.
Next one.
Yeah, please.
There we go.
So here what the artist has done is kind of pick out a bit of the retina, and you can see that it's hard to tell, but the retina has 3 parts. If you look at the image on the very left of the gray non-colored image, you can see the 3 spots. And here we're seeing just the middle part of that retina, and there's 6 parallel rows of receptors. You can count them top to bottom. There's 1, 2, 3, 4, 5, 6. They're called ommatidia. That's their names. And those are the only parts of the eyes that see color. So just that mid-band strip that runs around it like a tire tread around the middle of the eye is the only part of the eye that has color vision. And of course, that only looks at objects when the animal is looking straight at them. It doesn't see color in any other direction.
That's so weird. That just seems so strange that it only has like that little.
Yeah, they just have a strip of color vision in this whole world of black and white.
Yeah.
Basically. But they see many, many colors in that strip of color vision. And so many of those eye movements that we just saw in that last video are to bring that color vision system onto something they want to look at closely. And so they'll sort of scan over it to see what its colors are.
So do they, when they're kind of going along hunting for things, do they just sort of move along very slowly and kind of check things out?
Yeah.
Carefully before they decide if they're going to try to eat it, or
pretty much what they do. They'll move along. They'll often make little darts along the bottom and then stop and kind of scan the world around them to see what they've got. Or some of these species will actually hunt from holes and stick their heads out and then scan the world as it goes by, looking for things that are interesting. And if they recognize them, what they do at that point typically is they bring both eyes to bear on the same object and sort of estimate how far away it is. And then they'll decide whether they want to strike at it or not.
That's sort of like a chameleon, where they could kind of like turn their eyes to focus.
It seems to use the eyes somewhat like a chameleon. The only difference is that a chameleon, of course, doesn't have this triple vision in each eye. It only has single vision in each eye. And these eyes can't focus. They don't have a focusable lens, so they're sort of fixed focus eyes, unlike the chameleon's eye, which can focus on. Right. So the chameleon tells how far away something is, partly by focus distance. These do it by triangulation using the 3 parts of the eye.
It's so cool.
I think the next slide, there's a few more labels on this slide. We can just pop through them.
Oh, yeah, I see it.
This just shows the mid-band where we are. And then this shows you where the colored apparatus is. There's filters in the eye that create color vision.
I understand.
There's also part of the eye that's used to see polarized light, which we can talk about later if we want to.
Yeah.
Right, that they can see over a wider range.
Yeah.
Than we can.
Yes, they span a wider part of the spectrum because they have not only these color receptors, but they also have ultraviolet light receptors, which we don't have, so they can see in the UV. Yes. So here's again the sort of a graphic showing the trinocular vision. So the left is the whole eye, and the right is a view of just the retina and the optics. And then on the far right is the color vision system, which kind of visualizes just this array of colors in the middle. And the spatial vision that has these crossing over axes that allow the animal to triangulate how far away something is when it's looking at it.
Is there any other eye anywhere in all of life that is close to stomatopod eyes?
No.
That was bad.
No, really, I mean, there are other, of course, many crustaceans have compound eyes, but there's nothing that makes vision like this. It really, they went off on this evolutionary branch, and they kind of wandered into a new evolutionary universe that nobody else had ever been in before. And they built these, during that time, these eyes got built.
It's so amazing. And how does it help them, say, like when they're being stalked by an octopus?
Ah,
They do
well,
this.
hopefully if they're lucky, they'll see it at least and duck into a safe spot. I mean, it's interesting because they will prey. Their species, like the one we saw earlier, the peacock, which when it's a small animal, it gets preyed on by big octopuses, and when it's a big animal, it preys on small octopuses. So it's a sort of a bi-directional predation, depending on how big each party is in the battle.
I remember Mike Degree in his Incredible Suckers movie. He had a series where the stomatopod was kind of like going around and backing up through these little crevices and things, and the octopus was kind of going the other way and sticking its tentacle out, trying to figure out where was the stomatopod. And then it was an epic little soiree, you know.
So who won the war?
I think I've got to go back and remember, but I think the stomatopod got away.
Yeah, I mean, of course, octopuses are very good at dealing with cracks and crevices too, but they also have to deal with the smashers. So they can't take too big a risk in going.
That's right.
Like
hit the tentacle with a smasher would probably be pretty painful.
Yeah, I mean, the typical trick of an octopus is just to completely envelop the mantis shrimp, so it can't. It's like basically the tar baby at that point. It can't get out.
Oh, this one's got some embeds too here. So you got
It just shows you that mid band with all the color receptors and polarized light receptors. And the sphere there just kind of recreates how the top half of the eye sees the upper part of the world, and the bottom part of the eye sees the lower part. And then this mid band, which looks like this tire tread, just patches off a little bit in the middle of that, and where all three parts of the eye share a field of view.
That's cool. It's kind of like you would see in the water column anywhere. You have a kind of lighter, more light coming in from the surface, and then you've got it.
Yeah, that's interesting.
Yeah.
So they do have different adaptations for that as well. So this is the animal we work on the most. And here it's posing very sweetly, so you can see all six of the parts of the eye that are looking at the camera simultaneously. But it's not, when you pick up even the eye of a crab or even a honeybee, if you turn the head around, you'll see that there is this dark spot that kind of follows you no matter where you move the eye. But it's not really the animal looking at you. It's that's how compound eyes are built. If you just picture a box of soda straws, if you look straight into it, they look dark. But if you look from other angles, they don't. You're basically looking down these photoreceptors. And so the reason that's why they look dark. But of course, other parts of the eye are looking other places at the same time.
This is such a beautiful animal.
It'd
be a great to have in the aquarium.
Yeah, they are great.
The reef.
Yeah, they're favorite pets of my grad students because they're always busy doing something.
It looks like something you'd see at the Explorers Club.
Oh yeah, like a little brooch, you know.
The whole costume.
The whole costume, yeah. I'll dress as a mantis shrimp, have this sort of little shell looking.
Or maybe at the Oscars.
Yeah.
There have been a couple of movies made with people dressed as mantis shrimps, so there you are. So we're back to how the eyes vary with depth, and mantis shrimps also have to cope with the fact that some live in very shallow water, some live in moderate depths, and there's a few species actually that live in the very deep sea and still have eyes, and we're not really sure what they're looking at down there, to tell you the truth.
Luminescence.
So what's the deepest depth you found a mantis shrimp?
Well, I don't get them from these depths, but they've been reported from down to 4,000 meters, so they're really far down.
That's
those animals are called, their scientific name is Bathyspila. You could get the bathy referring to the deep sea, and they have good compound eyes. And again, it's like this. There's a fish down here on the bottom on the left side that has large eyes. You see large eyes in some of these benthic animals, and at this point we don't fully understand exactly what they're looking at because in principle there's no light. There could be bacterial bioluminescence, or there could be hot water luminescence. There could be what's called triboluminescence, which is luminescence produced by rubbing of things together, or sonoluminescence, which is caused by bursting cavitation bubbles or other kinds of loud sounds. We don't know what they're seeing.
That's gonna be fascinating, because if when we have submersibles down there, you're able to kind of just sit and watch and see what happens. But we see
Yeah.
so much bioluminescence all the time. It's just like this light show, but it's
Yeah.
really hard to see who's making the light,
Exactly, because if you turn the lights on, you don't see anything.
You see not anything. Yeah, 'cause, to your point, there are so many that are transparent or where they just flee from the light.
Exactly. So this is just a kind of peek here. So this just shows you that animals, these are fish retinas, and fish retinas have cone receptors like we have. Their color receptors are cones. And these colors in these pictures refer to the kinds of cones they have. And these are different species. But the point is, these are from different depths. And you can see that the open ocean ones or the continental shelf ones have just blue and green receptors. The coastal water ones that live in shallow water or near shore often add in yellow or orange or even sometimes reddish receptors. And then fish that live in freshwater where they're near the surface and the water is also very brown or reddish because of tannins in the water. They have red receptors, more like we do. So the retinas vary from one spot to another depending on how deep you are. And the same thing is true of mantis shrimps. I can't really give you a picture like this for mantis shrimps because we don't have a proper graphic. But the same is true that mantis shrimps that live in shallow water tend to have a much wider array of color receptors than mantis shrimps that live in deeper water.
Yeah, it would be sensible. Yeah. Oh, wow. What's going on here?
So here we're looking at the midband of a mantis shrimp. This is a picture by my former grad student, Mike Bach. Mike is now in Sweden. He still loves mantis shrimps. So it shows the 6 rows of this midband. And what Mike has done is this is being photographed with a backlighting of red light and a forelight of ultraviolet light. And in these eyes, you can see that there's a number of fluorescent molecules that produce this fluorescent color when they're excited with ultraviolet light. And this was the first time we really realized that there were substances in the upper retina, not in the photoreceptors, but in the actual optics that were somehow absorbing ultraviolet light. And that gave us the hint to look for ultraviolet filters in the eyes of the animals. So these animals' eyes contain a number of filters. I think they're shown in the next slide. So these are examples of the color filters. We already knew about these before. So if you look at the bottom row of pictures, those show up photoreceptors of mantis shrimp seen from the side. So light would travel from top to bottom in these, would come in from the lens, which would be out of the picture at the top. And it would go through the filter, and that filter would change the color that's coming out the bottom of the filter, obviously. And so that would be a way of tuning a color vision system. And the top row just shows you the same thing, but from the top. So you can see the red filter from the top or the yellow filter from the top. And when we first discovered these, we thought, the color vision of mantis shrimps is made by having all these color filters, that they just filter their photoreceptors, and that makes them sensitive to different colors of light. That was only the first step. It turns out that's only the simple part of the story. Also, each receptor inherently also has a different kinds of biochemical in it that responds to light of different wavelengths. So it's tuned both by filters and by its actual chemical photoreceptors. It's extremely complicated. And again, quite different from anybody else. So we have 3 kinds of color receptors. Mantis shrimps have 12 kinds of color receptors as a result of all this tuning. So in a sense, maybe 12 primary colors, although it's not really what they have, but they have 12 sets of receptors anyway.
I don't know what they must think of us when they see us coming over and we've got, like orange fins and we've got a blue suit and we've got a white snorkel and, all kinds of stuff going on.
And look at these images and Picasso, eat your heart out.
Right?
These are pictures taken of animals that have just been fresh frozen and then cut. So these are living pictures. They're pictures of actual what they look like in life. They're not stained in any way.
Amazing. Like little resistors.
Yeah, when we first saw these, we were stunned because this is not in any other eyes either.
Wow.
Not the unique mantis shrimp property. So in the end, you get this very complicated visual system. So this just shows the color receptors, and you can see this only shows 11 of them because we had one of them was not characterized when we made this picture. But you can see at the far left you have the ultraviolet ones, and then on the right side you have the ones that correspond more to our spectrum of light. And so the receptors kind of march through the spectrum. Now here we're looking at polarized light. So we don't think about polarized light much in our world, because humans are not aware of polarized light. We can see it in the sense that we see the light, but we don't know it's polarized. Just like a colorblind person can see objects, but they can't tell that they're colored.
Right.
So this shows a photograph underwater taken through two different polarized filters. And then if you put this through a proper analytical system, the next picture shows what happens when you process it.
That's
So-
pretty sweet.
So it allows, if an animal has a brain that, like a mantis shrimp might have a brain like this, it has polarized light receptors that see polarized light and would allow it to recreate the sort of full color image of the world by just processing slight differences in polarization that you saw in those first two pictures. And so you can see now we're getting much greater depth of vision than we had before. And we're getting much more color clarity than we got before.
So you're able to
This is
get
probably one way they use polarized light. Is
so
just
you're getting both the quality of the light, the color, but you're also able to get a better depth perception.
right. So it would give an animal a way to see deeper. To do that, you need a lot of brain power. So here, everything that's purple in this picture is brain. You can see that the central brain is at the bottom. It's that trilobe thing that looks like almost like a letter Y or something upside down. But all the purple up above is in the eye, is in the actual eye stalks of the eye, parts of the eyes, and you can see that actually it's more brain in its eyes than it has in its brain. And all that processing power is there to kind of extract information from this very complicated retina, which is the green parts at the very edge of the eye is where the retina is located.
That's just wild. Think about
They
it.
have a lot of power there. So I think the next picture just shows it from a different point of view. Again, you can see everything green here now is brain, is nervous system. The red stuff is muscles to move the eye around.
I was
That's
gonna
what gives
say.
these eyes this incredible mobility. And the retina you can't really see in this picture is way out towards the left edge where you can see the cornea, which is blue color in this picture.
But
It's a really nice image, I think, to show how much of the eye socket is filled with nerve processing power.
that's an amazing, proportion, because I mean most animals probably don't have that sort of I don't know.
No,
brain.
our retinas are very powerful processors, but they're very thin and don't have anything like this kind of complicated nerve tissue in them.
Diversity in invertebrates, we just get so accustomed to what our senses are and kind of think that's the way all things operate. But like an octopus has sensors like a brain in every one of its arms. Not exactly.
Yeah, it has more brain in its arm than it has in its head. And mantis shrimps have more brains in their eyes than they have in their head, so it's kind of a parallel there.
But the brain in the eye that's another whole dimension. Look at this creature.
Yeah, this is thanks to everybody who helped out with this work. So if you kind of page through the slide, the names of people will pop up. So these are members of my lab here that have worked on mantis shrimps over the years. And it's been a really great group to have. It's been a fun and a wonderful group of people. And then the next bit shows our international collaborators and people who've helped out with the work.
And so how did you get started being curious about the mantis shrimp?
I mean, hooked on mantis.
Hooked on mantis.
Hooked on mantis shrimps. It wasn't literal, thank you.
Good.
I was a grad student at Duke at the Marine Lab there. And we were out doing our invertebrate zoology class, and we were trawling for just whatever would come up. And up in the trawl came these really strange creatures that had these long claws like praying mantises, about 6 inches long, these animals. And I had no idea what I was looking at. I'd never seen anything like this before. And they were mantis shrimps, obviously. And the shrimpers get them all the time. The shrimpers call them finger poppers because of the way they behave. So I thought it'd be really cool when I became when I finished my graduate days, where I worked on crab larvae when I was a graduate student, which was also very cool. But I got interested in the eyes of animals while I was doing that, and I realized that mantis shrimp eyes were very different from any other eyes that we knew about. And so when I became an independent researcher at UMBC, I thought, well, I'll send them a grant proposal off to NSF to see if they were interested in having me study the eyes of these animals. And NSF happily funded my research right from the start.
Excellent.
And my thought was that they must have color vision because they're so colorful. At the same time as my work, there was a guy in England named Justin Marshall who was working on mantis and he discovered their filters and that they had this strange color vision. So we joined forces and he came to my lab in 1986 or 7. And we did all the spectrophotometry of the eyes, trying to work out how they sense light. And in about 2 months of hard work, and mostly in the dark, we kind of solved the problem of all these photoreceptors. And we never stopped working together after that. We still work together now. That's been a 40-year history.
That's so awesome.
So question, when you have animals in your lab, do you name them?
The grad students will name, there's usually one animal that lives in the tank in the lab. And the first one was, the animal we work on mainly is Neogonolectus ursidae, or NO. So the first one was Dr. No. And the second one was Mr. Googles because of his eyes. And the third one was called Waldo because we could never find it. We would come in and say, "Where's Waldo?"
Where's Waldo? See?
so that was the name of the three that were most popular in the lab. They all had their various names. Yeah. But the ones in the tanks that were used for research, I'm afraid we went through dozens of those and they were all anonymous.
Aw, that's it.
So they never got any fame. Mr. Googles got real fame because he was actually featured in National Geographic. So he did get international fame from Ed Yong, who's a science writer who was working on a story on eyes in the ocean. Came to our lab.
But they do behave differently. I mean, they're no two exactly alike. And sort of irresistible to give them names.
It is. Yeah.
It is true. It is true. But we usually we have one of each species. Usually you don't put more than one animal in the tank anyway, because
No.
the likelihood is one of them is not going to last very long.
Yeah.
You know,
we kept a damsel in the tank whose name was Bait. And that animal survived 3 generations of mantis shrimps. I don't know what he had going for him.
Speed.
But Bait outlasted all the mantises. He was still alive when the tank has actually been shut down now because my lab is closing down, but it is now gone.
Yeah.
I had the pleasure of going to Roy Caldwell's lab at UC Berkeley.
Yep.
Meeting his pair of mantis shrimps that had to be relocated from a glass tank to a plexi tank because they he came in one day and found that the tank was totally empty of water and had a divot in the side where the mantis shrimp had knocked out a hole. Luckily, the hole was about 1 inch above the bottom of the tank, so there was just enough water to keep the mantis shrimp alive until they could get it into a new tank.
Wow.
But some of them do live a long time, and don't they say mate for life, or at least pair bond for a while?
Yeah, the species, the spears, like that we saw early in the slide, that's a species called Lasiosquilla or Lasiosquillina, depending on the species. They live in burrows in mud, in mangrove mud, and they will pair off for very long periods of time. We don't really know how long the pairs are stable, but probably for years.
Mm-hmm.
We do know that if we catch them with a noose, so you put a noose down into the burrow and you entice them up with a fish, and then when they come up, you can noose them just like you would noose a snake or something and pull them out of the burrow. So that leaves that's always the male that hunts. The male's on top and the female's down making eggs. And so the female now is a bachelorette, or a widow, I guess is a better word. And within a week, no more, there's a new male in that burrow. So there's a lot of traffic going around looking for burrows, and we don't really see it, but we know it happens.
I would imagine so.
Hanky-panky among the stomatopods?
Yeah.
Well, I'm afraid so. Well, it's driven by us who pull out, remove some of the piece of the population, unfortunately.
I guess the males would be kind of susceptible to things like, egrets or other birds
They
that
are.
have good vision and can kind of spear in and grab them from the burrows.
Yeah, I had never seen it myself, but Roy has encountered animals in nature that are missing an eye.
Ooh. That would be strange. It's like one going around.
Yeah, but well, one eye can see everything, so it may not, it may not be
May
that
not
much
be
of a
that bad,
handicap.
yeah. All right, we've got a couple questions that have come in.
Okay.
Caroline is saying, thank you for doing this. So if colors aren't visible in the deep water, why do you think there are so many differently colored organisms down there? I'm amazed at the beauty at the hydrothermal vents. And 17 years ago today, I dove in the
We
Alvin.
lost the feed for a minute.
Yeah. Oh, you there?
I missed part of that because you froze.
Okay.
Could you
She's
repeat it?
asking, if colors aren't visible in the deep water, why do you think there's so many different colored organisms down there?
Okay, well, we talked about this earlier, strangely enough. So once you get below 1,000 meters, it's dark, and it's permanently dark except for bioluminescence. But fish in that animals living in that depth, if they're fishes, they're typically black, because black absorbs light. In fact, it's been shown by my colleague at Duke, Salka Johnson, that the black of black fishes is as dark as the darkest black we can make artificially. It reflects essentially no light. So if you have black paper reflects about 5 to 10% of the light. It looks black to us because everything else is brighter, but it's not very dark, really. Fish really reflect about 1/10 of 1% of light. It's essentially nothing comes back. So that's one thing. Of course, that's black. Most crustaceans are red because in the deep sea there's no red. So as a result of that, they're just the same as black. Many animals in the deep sea are transparent because, again, if you're not black or red, you can be transparent and still be invisible. You don't see other colors in the midwater. On the bottom, however, like we talked about briefly earlier, there can be some coloration down there, and we don't really, at this point, know exactly what the illumination system that is being used for that is. We know that hydrothermal vents emit infrared light, but that's still not very useful for color vision.
Yeah.
You know that above this 1,000-meter limit, once you get up into 600 meters and above, you get lots of colored fishes. But you don't see that much color in the deep sea itself.
Yeah, even like the sort of the Yeti crab is, some of them look kind of yellowish, but they're most of them are kind of really pale.
That's one of those interesting benthic exceptions that I just was talking about. You
Yeah.
don't see that in mid-water though.
No.
But the color may just be incidental, not functional.
Yeah, well, there's no light. It may not matter what color you are.
Yeah.
Got to be some.
The reason why mid-water animals are black is not because of downwelling light. It's because of bioluminescence. They don't want to. They don't want to be seen to a bioluminescent searchlight.
That's right. Yeah.
And some fishes have evolved red bioluminescent searchlights and probably use them to search for crustacean prey.
Wow.
Although we don't know, we've never seen this because we've never observed it, but it's a hypothesis that makes sense.
Yeah. They
could also use it for secret signals to each other because they can see the red bioluminescence, they can see each other as well. So it may be a way of finding mates.
Secret signals. Yeah. Okay. Emily is asking, evolutionarily speaking, do the mantis shrimp share common ancestors with terrestrial insects?
Well, yes, they do. I mean, everybody shares an ancestor somewhere, but the crustaceans and insects are closely related. Crustaceans and insects are on the same. There's many animals that have hard skeletons. There's spiders, there's scorpions. Well, that's the two other main groups. There's the crustaceans and insects. Crustaceans and insects are actually on the same branch of that tree, so they actually are sister groups. Mantis shrimps probably split off from everybody else at least 400 million years back. So it's been a long split. The other insects probably split off from the crustacean mainline about the same time. So they're sisters, but they're sisters for 400. A common ancestor is 400 million years ago. It's a long time.
Like horseshoe crabs.
Yeah.
Yeah, same thing.
Years ago.
Horseshoe crabs are more related to spiders than they are to crustaceans, however.
Yeah, but the whole arthropod origin.
Yeah, they're also arthropods, and they're also a different branch of relatives, but more distant than insects are.
Yeah.
Right.
Doesn't the horseshoe crab have the 8 eyes too, like a spider, right?
Horseshoe crab has a pair of compound eyes and a pair of accessory compound eyes. It has 3 eyes on the front. It has an eye, and it's underneath of its shell in the middle. It's called the ventral eye, and has a series of photoreceptors in its tail. So I don't know how many I've said so far, but there's a lot. And And yet it's, its brain is not very competent. Its behavior is very simple, but they have extremely good vision. I know one time when we were out hunting for larvae at night, in a pitch black night, it was the same night that Limulus was hunting for mates, and we were wearing black booties, and the males would crawl over our feet. No matter where we stepped, we'd immediately be attacked by a male Limulus. So they could see us in light that we couldn't even see anything.
Wow, that's incredible. Okay, so Maria she says she's one of Ray Caldwell's early grad students at Cal working on stomatopods.
Okay.
But let's see. One of the spheres had a functional morphology with the eyes clearly has something to do with their complex communication. Can you comment on how the eyes are used for communication?
You mean like blinking, winking?
Blinking.
Yeah. So the spheres tend to be pretty colorless by and large. You saw the example we had. It was mostly just dark and white, maybe a little bit of yellow on it. Even though they have good color vision, the animals that seem to use colors for communication are the smashers, and they have a number of colored markings on their bodies, and some of which they display. They show off, like their elbows when they pull their elbows up. I guess I can kind of do that. They show off these patches that are here on their upper arms that are used for communication. And those patches are colored that match the species that they are. So it varies among the species. And so it's a way of recognizing what species you're talking to or fighting with when they make that display. And so that's probably a very important signal. They also use polarized light as signals. They have polarized light patches on their bodies that we can't see. I mean, we see them, we don't know they're polarized, but they can recognize as signals as well. So the interesting thing is that the colors of the animals' bodies are not matched in any particular way to the colors of their visual systems. So most manuscripts have very similar color vision systems. They only vary with how deep they live. But their colored signals, of course, vary a lot. But it's like birds. Birds have basically the same color vision system. Almost all birds see the same color set. But think of all the variety in colors of birds. Each species is signaling its own self by its color patterns. And I think manuscripts are doing the same sort of thing.
Yeah, it's pretty fascinating. And how do the manuscript, do they sort of, like some birds will kind of shift different parts of their body so you can see the colors more like a
exactly. A manuscript is the same sort of thing.
Yeah,
exactly right. So they'll display the parts of their body they think are that they think. I don't know what they think. I suspect they do think. But anyway, they display the parts of their body that are the most likely to be attractive to or in the case of aggression, most scary to whoever they're looking at.
So they can sort of use the different little appendages and
exactly
smashers and elbows and things to just sort of reconfigure.
And on the sides of their face, they have these things that are called antennal scales that they can also move like this to display colors when they do that. You'll often see them doing that in a face-off. That's an additional signal they can use. It's also polarized, by the way.
Polarized, yeah. Well, we're just about at the top of the hour, and I just really wanted to thank you so much for making the time to talk to us about how animals see. I hope everybody goes home and kind of contemplates their dog or cat and thinks about, how does he really see me?
Yeah, it's been a pleasure to work with you. It's really been, I really thank you for inviting me to come.
There's a new book with a big section about stomatopod vision. It's The Immense World.
Right, Yed Young wrote that. And he's the guy who wrote about Mr. Google's as well.
Oh, cool.
And he talks a lot about our lab in that section on manuscript vision.
Yes.
Yeah. Well, I hope everybody goes out and
Immense World's a great book, well worth reading.
Next animal.
It's about sensory ecology, which is what we do.
We've got to think about how those little bees out in the garden and the spider in the bathtub and how they all look at you.
It's something really different. Everybody sees us in a different light, so to speak. It's
really amazing. Because lots more reasons to remain curious, which we should all strive to do. Yeah. And before we close, we'd like to say thank you again, Tom. Thank you to our producer, the Ocean Elders. And mostly, thank you everyone in the community who shows up and dives in with us.
Yeah.
We're so grateful to all of you. We're going to be back next month. We'll be talking about parrotfish.
Yes.
Okay,
Yeah,
I'll be there.
thank you. And until then, remember, take care of the ocean
As if your life depends on it.
because
It does.
it does. Thanks again, Tom. We'll see you everyone next time. Bye
Thanks.
for now.
Have a great new year.
Thank you.
Bye-bye for now.
Bye-bye.